Right. Good morning. Thank you for the invitation. I am here to talk about a virus who is infecting a parasite that is infecting the human host. The parasite is this, maybe some of you know, are familiar with this parasite, is Leishmania parasite, an intracellular protozoa that group a big amount of species that are associated with different type of manifestation. Maybe, as many of you know, Leishmaniasis is a complex disease, and I'm going to concentrate more about the cutaneous manifestation of the disease. That is the one that is predominant in Colombia and in many places in South America. In Colombia, we have about 10,000 new cases per year, and that is important to note that most of the infection, when the people is infected with Leishmaniasis, most of the cases are totally asymptomatic. In different type of studies that we have done, we have 70% of the population that have been exposed without any symptoms. But for those who develop the symptoms, as you can see in these type of lesions, they are extremely complicated lesions that are not self-healing. So we need to treat these patients, and the treatment that we are using at this very moment are systemic treatment. So one early complication that we have for cutaneous leishmaniasis is precisely the treatment failure. Many of these patients do not respond to treatment, or they do present relapses after 6 months being treated. In Colombia, and in other South American places, we calculate that almost 25% of the patients present this therapeutic failure. But this other complication of cutaneous leishmaniasis is probably less frequent. There is no reliable data about, but we calculate that about 10% of the patients that suffer for infection with Leishmania brasiliensis, one of the predominant species that we have there, develop this late complication known as mucosal leishmaniasis. This complication can appear 10 to 20 years after the primary infection, and is a highly deformed disease, and many of these patients, they even do not consult because it takes so long for them to realize that this is what's happening inside these mucosas. So there are many species producing the disease, but in South America, the predominant group of species related with this disease are species grouped under gender and the subgender Viania. This group of species are more prone to present the previously mentioned complication. They are more prone to present therapeutic failure as mucosal leishmaniasis. So reasons for the complications are not well known, but recently it has been produced some important evidence given a role for this virus. This is a double RNA virus that is present in the cytoplasm of some strain of Leishmania parasites. This virus has been studied in some animal models, producing important evidence. Basically, what this experiment showed is that when the animals are infected with parasites holding the virus, they develop an exacerbated immunological response mediated by interferon, and as a consequence of this exacerbation in the immune system, the parasites tend to persist, and they present more risk to relapses. Also, in animal models, it has been shown that Leishmania parasites that hold the virus develop metastasizing capacity that it is related directly with the mucosal lesion. This same parasite, when have been cleaned from the virus, they do not develop the metastasis. At a clinical level, the evidence is not that strong. We have a couple of studies showing that when the parasite is infected with the virus, we have a more therapeutic failure in patients that suffer from cutaneous leishmaniasis, when the species of parasite is Leishmania brasiliensis or with Leishmania guyanensis, both of them belonging to the Viania subgenus that I mentioned before. At the epidemiological level, the evidence is highly variable. And there you find that the strength of the association between the complications and the presence of the virus is variable between regions and between studies. So we particularly thought that this variability can be explained for the fact that the detection method is not robust enough. So trying to make a more systematic detection approach, we use all the described PCR protocols. They detect different regions of the virus, and first of all, we take into the consideration the type of sample that we are dealing with. So when we are dealing with isolates, we do a simple PCR using as a target this region of the virus capsid, and we do a calculation of the parasite load using a different region here corresponding to the 5' UTR. But when we are dealing with biopsies coming directly from the patient, first of all, we do the detection of the parasites, and only in cases that the parasite is positive, we do the detection of the virus through these different regions. It's a protocol described by Ramos, and there it's important to note that this protocol corresponds to nested PCR. When with this type of detection, here I have an example of the detection that we do with isolates, with parasites that have been isolates. And basically what I am showing here to you is that with this approach, we were able to detect very low very low viral load, as low as 410 to the -1. And this is a simple PCR. When we do the detection with biopsies, we have a better result. I'm going to show you. So here is the percent of detection that we do in our series of patients. About 47% of the samples corresponding to isolates hold the virus in our patient, that all of them were complicated outcomes. And about 60% of the biopsies hold the virus. However, for these patients that we have two types of samples, we have the isolate and the biopsy from what the isolate come from. And this is the most interesting result, because for those type of patients, we found that 50% of the isolates that were negative for virus detection, become positive in the biopsy. So what it means is that the virus is lost during the in vitro growing. That the results then increase the sensitivity of the biopsy up about 70% of the samples. So basically, the message that I have for the panel here today is that in order to do studies that really are able to find the role of this virus in the complicated outcome, we need to look for the virus in samples coming directly from the patient. And in that case, we were able to do it in our research lab with a simple and cheap method as RT-PCR. But this is not a robust method. Maybe many of you are familiar with that, because we have a lot of problems with cross-contamination and all the high technology, etc., that have been saying here. So this virus needs to be detected because, first of all, we need to address the question whether or not the virus is involved in the complication of leishmaniasis. And secondly, in case that it is involved, now we need to prepare a true detection method for the virus, because the virus needs to be treated at the time that we treat the patient. So that's basically the message that I want to leave with you today. This work was made basically by people from my lab, Marcela Parra and Samantha Ponte, in collaboration with people from the Army Hospital in Colombia and people from the Reference Center for Leishmaniasis, for treatment of Leishmaniasis. I would like to give special thanks to Professor Beverly from Washington University, and thank you for your attention and your patience with my English.